So in this last bit of this lecture, I just wanted to show you quickly what a real instrument looks like. So what I've drawn you on these paper diagrams are very simple, objective and tube lens is all we need to discuss. And here I show you that what a real microscope looks like, and it looks a whole lot more complicated, but in fact it's the same thing. So here you have on the left-hand side what is a modern research microscope, and it has lots of lenses in there, but the really important ones are just the objective, which sits here. Can you see that? Yes, I drew that in. Yeah. So here's the objective, and you have the tube lens. So the tube lens will be probably here. And if you look by eye, then you have a tube lens and also an eyepiece in the sample, and then you put your eye there. And so you can direct the light in different ways. You can excite. So let me just get rid of these again. I can illuminate the light from below to do a bright field microscopy experiment. So the light goes in from down here, bounces up, goes through the condenser onto the sample, is refracted and diffracted, and is then captured by the objective lens, passes through this, and then goes up here, goes through the tube lens, goes up here, goes through the tube lens, and forms an image on the CCD chip here. And if I slide in the correct piece of optics, I can also direct the image, the light, onto an eyepiece. So it goes through, there's a tube lens here, and then it goes through here and onto my eye. And you can observe the bright field image that way. So that would be bright field. This is an upright microscope because the objective sits on top of the sample. The sample is down here. There is a focusing knob and a sample a specimen stage, and the focusing knob allows you to put the sample into focus with respect to the objective to get a sharp image. This microscope can also be used in fluorescence. And in fluorescence, we excite and detect through the objective. So what you do is let me just change the color if I can do this. I'm not sure I can. I don't know what's happened now. Let me just try it one more time. Yep. So if I take blue light for this, let's suppose I have light coming through. It goes now through filters, so I have the right color to excite the molecules that I'm interested in. So here's the dichroic that I showed you earlier on. The excitation light goes through the objective onto the sample, and then the sample fluorescence is redshifted. So it will come through. It passes through the dichroic and then goes through the tube lens, and you form an image on the camera chip. And usually there will be some filters in there as well, the emission filter. Alternatively, again, you can slide in this prism here and then send the fluorescence signal onto your eye if you want to do that. And again, so you can see that this is an upright microscope. This idea on the right-hand side about conjugate planes in the microscope, I do not want to talk about this right now. This will come back at a later stage, and we'll go through that. And with that, this lecture is done, and I'm looking forward to seeing you in the next lecture, where we will begin with quantitative aspects of imaging.